We are recording. Let's do it. All right, so starting with kind of key questions, we're going to start with the blood and immune system. So hopefully we all know blood and immune is still on this exam, even though that felt like it was a while ago. Hopefully we've already kind of looked at all the key questions and kind of have an idea of what he wants you guys really to understand. So starting off, let's look at this first one. It's going to be components of blood. So if we're looking at a tube of blood and we filter this down, so we spin it down with a centrifuge, then things are going to kind of layer themselves based off of weight and components. So kind of knowing what's in each section is going to be important. Starting off on the top, thin layer here. All right, so yellow layer, what do we have on the top of this? So when we centrifuge it down, what actually settles at the top?
Plasma.
Yep, so this is our
Plasma.
And what's in the plasma usually? What would we find?
Proteins.
Yep. So proteins. We'll have solutes, so like ions, sodium, chloride, things like that. I think another really interesting thing to point out is you would probably find antibodies here because a lot of the time we have plasma B cells, and those B cells secrete their antibodies into the plasma. So you might find some antibodies, which would be kind of under the category of proteins. A lot of this is also just going to be water. I'll have some water in there. But that's going to be what's located at the top. This middle layer is going to be our Buffy coat. And within our Buffy coat, it's going to be our white blood cells. So this is where we're actually going to find true cells. So cells that have a nucleus, cells that work within the immune system. Another thing is our formed elements. Formed elements are going to be like our platelets, which we know work with blood clotting. Oh yeah, go ahead. I wasn't sure if someone had a question. And then the bottom here is going to be really what makes up blood, which is our red blood cells. So this is our hematocrit. So our red blood cells or erythrocytes. Erythrocytes lack a nucleus, so we won't find, like, these are not true cells. They're cells, but they aren't true cells. They don't have a nucleus. They're just going to be essentially sacs of that hemoglobin, like what we talked about when we talked about respiration. So what they really deal with is just that oxygen transport from the lungs to the tissue and then transporting CO2. Cool. So that's our first question. Now, looking at proportions, what makes up most of our blood?
Red blood cells?
Most of it is water, yeah. So actually, if we really break it down, plasma is about 55% of the blood, and then the formed elements and cells are going to be about 45%. And then the rest, so we're talking about the formed elements and the cells, we're kind of including hematocrit in there. So a lot of it is going to be really water. It's like 92% of the, this is like very specific. 92% of the plasma is just water. And then really you only have about 7% proteins, and then 1% of that is other solutes. So that would be your sodium, other ions like that. So 7% is proteins. 1% is the sodium or other solutes. And then within the Buffy coat, really your blood cells, white blood cells are going to be more or less than 0.1%, along with the platelets. And then erythrocytes do make up the majority of it, which makes sense because we're going to want to get oxygen throughout the body. So a lot of the blood is going to be made up of our red blood cells. So it's like 55% total, and then the rest is about 45%. Cool. Any questions on components of blood? Well, if not, what is the structural and what are the structural features and functions of red blood cells, white blood cells, and platelets? All right, so when we look at red blood cells, which we call erythrocytes, their main function is to carry oxygen, and they do this by using hemoglobin. Hemoglobin is a 4 subunit protein. It has a porphyrin ring in it, and it has 4 iron atoms that are actually where those oxygens are able to link and combine. So I think that kind of jumps back into a little bit of respiratory, which we could potentially ask you questions about blood that kind of relate to respiratory, since they kind of work together in that way. The other interesting thing with red blood cells is their biconcave shape. So they have a little bit of like they're almost fatter around the outside, and then they have a disc, they're almost like a disc-like shape. So they have a little bit of a opening, not an opening, but it's like a concave thing in the center there. And I think the really, really important thing is with erythrocytes is, and we'll get to it, is kind of their lineage of where they come from, and then how they're actually going to die versus how we're going to make more of them. Like those two processes are really important when it comes to physiology. Now, white blood cells, which are our immune cells, that's basically their function. They're going to act as immune cells. They're monocytes, which become macrophages. They'll be lymphocytes or leukocytes. So like our neutrophils or basophils, or those are granulocytes. But all of these derive from the same lineage. They all come from hematopoietic stem cells. So we'll go through that lineage and kind of what to look for there. But their big function is obviously immune support. So they're going to phagocytose things. They'll be antigen presenting cells. So APCs like macrophages or naive B lymphocytes, things like that. And then what was the last one that was on here? And then platelets, their big function. So platelets are actually little chunks of a cell itself. So a megakaryocyte are the ones that actually make platelets. So when these pinch off little pieces of their cytoplasm and put them into the bloodstream, they can act as clotting factors. Is kind of their big role. So they're going to go help with blood clotting throughout the body. So they can do platelet counts. Like if you're getting surgery, something like that, if you have low platelet counts, then they might reconsider so that you aren't at risk for bleeding out, because they want to be able to make sure that you can actually clot. Cool. That's a pretty straightforward question. Not too bad. Just kind of knowing the function of things. All right, so this is really where our physiology stuff comes to mind. For me, I think understanding these processes is important, but then being able to actually relay the idea of like, if we were to tell you about a disease that disrupts the function of something, then I want you to understand how it's going to have an effect downstream. So let's start with just the synthesis of red blood cells. So if we look at lineage, we start with the hematopoietic stem cell. And these stem cells, they can take 2 routes. I'm going to go this direction with the blood cells, but it can also take a route that is our lymphoid stem cell. And this is going to be more important when it comes to looking at the immune system itself. But since we're talking about blood, we're going to actually look at the other direction. So the other direction is going to be our myeloid stem cells. And to get here, we need a specific marker that tells the cell what it needs to do to get to that degree or get to that or differentiate into that cell. So this will be our multi CSF. Once we're here, then we can break into 3 other categories. Since again, everything arises from a hematopoietic, the first one will be our erythrocytes. And if we're looking at this, I'm going to break this down a lot further. Let me move these a little bit so we have more space. Just realized as I do that, I'm going to lose track. Okay. So we'll have progenitor cells are going to be one direction that will go. These cells are going to slowly either become blast cells, which then can differentiate into our granulocytes. So all of the phils, the basophils, the neutrophils, the eosinophils. So those are all of our granulocytes. That's where they're going to start to differentiate. So these can then become and luckily you don't have to know all these lineages, but we can just kind of generalize that this will go to our granulocytes eventually. Now the other pathway is it's going to become a megakaryocyte, which we talked about these a second ago. These will become our platelets. So these sit close to the bloodstream in the bone marrow, and they'll just pinch off little pieces of their cytoplasm into the blood that become our formed elements or our platelets. All right, now the most important one is our erythrocytes. So starting from the very beginning of how these erythrocytes become is from this myeloid stem cell, they're going to become pro-erythrocytes first. So we have to think about where they're actually staying. So they're going to become pro-erythrocytes. And this happens in the bone marrow still. So right now we're still located in the bone marrow. We haven't moved to the circulatory system yet. From there, we're going to become a normoblast. From a normoblast, we then differentiate into what we call a reticulocyte. And then our last one is going to be our actual erythrocyte. So our mature red blood cell. So what's important here is we have a certain receptor on our proerythrocyte. I'm just going to draw it like that. And this receptor is specific for EPO. EPO stands for erythropoietin. And erythropoietin is something that comes from the kidneys. We'll go through this whole process, but when this is initiated from the kidneys, it's usually initiated by the lack of oxygen in our arterial blood. So there's cells in the kidneys called periocytes. They're going to detect that oxygen concentration. If it's low, then we release EPO. EPO runs through the bloodstream, gets to the red bone marrow, and then it's going to tell these proerythrocytes to differentiate into their normoblasts. Once we differentiate, this normoblast is going to do something where it ejects the nucleus. Because remember, erythrocytes are not true cells. So we're going to get rid of the nucleus and other organelles. And really the reticulocyte houses just some ER. And I like to just say like chunks of ER. So chunks of that endoplasmic reticulum remain within the reticulocyte. And then from there, it's going to mature fully to become an erythrocyte. And at this point, we've entered circulation. So everything on this side is going to be in our red bone marrow. I'm just going to abbreviate to save time. So red bone marrow. And then erythrocytes have now entered circulation or circulation. Cool. So now to kind of tie this story the rest of the way, if we are looking at our kidneys, we'll have a renal artery and then a renal vein. Blood obviously enters the kidney through the renal artery. So when blood comes in, we have these specialized cells called periocytes. I don't think I know how to spell it, so that's my best bet. Periocytes. These detect oxygen concentrations. So if O2 is low in our arterial blood that came in from the right or left side of the heart, entered through the renal artery, our kidneys are like, oh, this is lacking oxygen. Then we're going to release that EPO from the periocytes. So we get EPO with erythropoietin. And then this is going to go act on our bone. And obviously, specifically the red bone marrow. So red bone marrow. It's going to act directly on the proerythrocytes, like we said up here. And then in turn, we're going to end up increasing our erythrocyte. Production, which then is going to lead us to our final little step, which is where now we have increased. I really actually shouldn't even put it that it's going back to the kidney, but technically the kidney will then detect that we now have, with an increased erythrocyte amount, then we've also increased oxygen concentration in the blood, because now we have more hemoglobin making its way around, picking up more oxygen from the lungs. So in turn, the big end all be all is we've increased oxygen. I think it is still important to understand the EPO can also just be released if we're just lacking erythrocytes in general. So this is just the actual way that erythrocytes are being differentiated from that hematopoietic stem cell rather than going down any of these other lineages. So they'll need that EPO in order to kind of become those proerythrocytes. And I did miss one little detail. There is another interleukin. So to get from the hematopoietic stem cell to the myeloid, you will have a interleukin here. Interleukin 3 is also playing a role to get us from the hematopoietic stem cell, myeloid stem cell, to the actual proerythrocyte, is we'll need that interleukin 3, which is just another communicator telling that cell what to differentiate into, so that it doesn't end up going down this line or this line, and we stay going down the pathway to getting to our erythrocytes. Okay. Next thing is understanding where they go to die, right? So if we're looking at, it's my spleen. It's a pretty good spleen, actually. Spleen has a really funny looking artery. So we have a splenic artery. You'll have a splenic vein too. So when blood enters the spleen, we have two regions. So we have like a white pulp, and then we also have what we call a red pulp, which kind of surrounds the outsides of it. This is not drawn anatomically, but and what does the white pulp deal with usually? Does anyone know?
Lymphocytes, right?
Yeah, so this is gonna be our white blood cells. This is where we're actually can monitor for like blood-borne pathogens, so pathogens that are inside the blood. Since the spleen is kind of looking at our erythrocytes and looking around, then this is where things can go and be essentially eaten up by macrophages or other white blood cells. So that's kind of what the white pulp deals with. Now the red pulp, that's why we can kind of call it an immune organ in itself, because it has those germinal areas where we can actually kill off things. But the red pulp is really what's important. So the red pulp deals with erythrocyte recycling. I don't even know if I should use the word recycling, but I will for now. So what it looks at is it's going to look for oxidized glycoproteins that are on the cell's surface, on the erythrocyte surface specifically. So when those cells come in, if they have been super oxidized on those glycoproteins, then we can look at it and be like, okay, this red blood cell is old, which they only live about 90 to 120 days. So once they're super oxidized, those glycoproteins are oxidized, then we can tell this erythrocyte that it needs to be phagocytosed. So if oxidized, then will be phagocytosed by a macrophage. So macrophage will eat it up. And then all of those leftover parts then go out to the liver where they can be recycled. So usually our bodies are pretty low in iron. It's not one of our, like it's not a nutrient or something we can make. So we try to recycle that iron that comes from the red blood cells in the liver. We can recycle it and reuse it. And then the other big ones are bilirubin and biliverdin. Those are the ones where I think he had a slide where it kind of talked about jaundice or just the buildup of those. So usually you can tell if somebody has a dysfunctional liver because their skin will turn yellow, their eyes will turn yellow. And that can kind of tell you that their liver is not working correctly because it can't break down the bilirubin and biliverdin, which those are the two things that come from breaking down the red blood cells. So a lot of the time newborn babies, because they have a gamma subunit in their hemoglobin instead of a beta, their red blood cells go and die pretty quickly once they're born, so they can make the new ones with the beta subunit. And when that buildup happens, you can have a buildup of the bilirubin, which then causes babies to have that infantile jaundice. But yeah, so that's kind of where we're going to be recycling our red blood cells, is through the spleen. Let me see if I missed anything else on this question. And then, yeah, that's that heme recycling. So anything that comes from there. So let's just add it on here. So the iron, let's do it in black. The iron, the bilirubin, oops. These guys are going to go off to the liver, where they can then be either metabolized, so the bilirubin and biliverdin can be broken down further and then excreted, or the and they're usually eliminated through the feces, so they'll be excreted into the small intestine and then released out the bowel, while the iron is then retaken back up to be put back into the red bone marrow to then be put back into erythrocytes. Then we can keep recycling that iron since we don't make that nutrient on our own. Questions on that? Cool. If not, I kind of want to go back to this flow chart that I already had going. For the next question, it's kind of talking about the differences where we regulate how we get to red blood cells versus white blood cells. So we have a bunch of different markers that occur, and I actually like the picture. Let me find the slide number it's on. Slide number 12 on the immune system, lecture 41, has a nice flow chart of kind of where I think you kind of need to understand where some of these things are working. So I think obviously this is a big one. This is the one that's getting us to that myeloid stem cell. Is there a question? Sorry, I'm talking too quick. Okay, so we know to get to the erythrocytes, we need that EPO. We have some interleukin-3 is kind of the big ones. Now from here, to get from a myeloid stem cell to get to the progenitor stem cell, we're going to have something called GM-CSF. So this is going to stimulate the production of our granulocytes. So those progenitor cells become granulocytes. I guess I could add an arrow. So we need that in order to actually proliferate into the progenitor cells. The megakaryocytes don't really have a signaler. They're just going to either, I don't know what actually persuades them to become from the myeloid stem cell lineage to the other one. I was actually thinking the same thing. Let me double check. I feel like GM stands for like growth something.
GM is going to be for granulocytes or monocytes.
Yeah,
The
macrophage.
that's gonna, yeah.
So granulocytes, so GM. This kind of makes a little bit of sense since you are becoming a granulocyte at that point. The other one is the MCSF, which is going to stand for the monocyte. But that's going to be down another. So yeah, this chart can be a little bit daunting, I won't lie. So from the progenitor cell, we also can have what we have, or we have a myo or monoblast. Monoblasts will become monocytes. And monocytes become macrophages once they're mature. So we could actually kind of look at this and look at, okay, so GM is granulocyte macrophage pathway. So we're going to granulocytes here from the progenitor cells. There's obviously a few cell lineages before that, but we're not going to worry about those. And then the other one is going to be our myoblast, or yeah, monoblast that becomes a monocyte. And then this one is going to be our MCSF to get to that lineage. Now for the granulocytes, I probably should add the 3 that you guys need to know. I just say the phils because it just makes it a little bit easier, but we have neutrophils, basophils, and then the eosinophils. Mute somebody here. Okay. And then to get all the way to granulocytes, I needed to add one more marker here. This will be our CSF. So first signalers are GM-CSF. Granulocyte macrophage CSF is getting us from the myeloid stem cell line to the progenitor stem cell line, or the progenitor cells. This then breaks into 2 where we have our GCSF, which gets you to your granulocytes, so our neutrophils, basophils, eosinophils. And then our MCSF, which we could say stands for the macrophage or monocyte, gets us to the monoblast monocytes. I don't think you need to really fully know all these cell lines. I think what's really important is obviously this first one, multi-CSF, GM-CSF, because that one's the one that really breaks you apart from either becoming erythrocytes or progenitor cells. So those 2 I think are really important to look at. And knowing that EPO is obviously the one that deals with erythrocytes is going to be important as well. As for the lymphoid stem cell line, there's no real markers at this point that you guys have to worry about that will get you over to that one. But we do know lymphoid stem cells become the lymphocytes. So we have our B lymphocytes and our T lymphocytes. So those will be their end-all be-all location at the end, or their end-all be-all differentiation at the end. Yep, that's right. So agranulocytes, I want you to think of them as they do not contain granules within the actual cell. So granulocytes like basophils, neutrophils, eosinophils, if you were to look at them under a microscope, you can see that they have these little granules in them, which is what gives them the name granulocyte. A granulocytes do not have those. So those lymphocytes that we talk about, so B and T lymphocytes, they don't have all these granules everywhere. And then I think really that's kind of the lineage breakdown. Do you guys have any questions on that? Or at least what I feel like is going to be important for you to know. Cool. If not, let's move on to the last two. So a lot of this has to do with the blood doping. We know that blood doping kind of deals with you taking synthetic EPO, things like that. However, it's found that actually doesn't really help. People believe that if you take EPO and things like that, then you're able to actually increase the amount of blood and oxygen you're getting to your muscles. But I think that's kind of the big key parts. I'm not going to write all that out. I think if you guys can just kind of go through that. So this is the EPO doping. So, so you can take synthetic EPO. This would obviously increase our O2 concentration in the blood. If we're getting more O2 within the blood, then we're able to actually get more to the actual tissue itself, which in turn, gives your muscle more oxygen to work with when it comes to cellular respiration or cellular metabolism. But obviously some of these risks, there's higher risks looking at that. I don't really want to go through the pathologies. I think you guys can kind of look at that yourselves. I just want to go through more of the processes that we have to know. So make sure you guys check out these questions if you haven't looked. Do you guys have any questions on blood? I feel like that's a lot. There is a slide I do want you to put some attention to. It is slide number, and really I don't need you to memorize everything that's on this slide. I just think it has some good information. It'll be just the 13. So it's right after that one with the big lineage, just kind of knowing how each one of these baso, eosinophil, neutrophils kind of work. Like I said, I don't have enough time to go through all of that, but you guys can check that out. All right, let's look at lymph. So what are the primary and secondary lymph tissue? All right. So lymphatic tissue obviously makes up, we have lymph nodes. These are going to contain germinal areas. And they help to filter out some of that stuff that came from blood, right? So we're looking at that whole idea of taking some of the fluid or things that are in blood, pushing that into the extracellular space, and then that is also pushing it into the lymph. The lymphatic vessels are then going to bring it through these lymph nodes where they can kind of filter out any sort of pathogens or things that it has found within the body or the extracellular space, so we can start cleaning that out. But we do have some specific lymph organs. So we have our tonsils. We're going to have our thymus. It's by the heart. The spleen. Your adenoids, which those sit kind of behind the nose. And then we do have some GALT, which is gut associated. these are going to be kind of located all around the intestines and things like that. So we will have some areas that focus on cleaning out lymph and are considered secondary lymph organs because they do help with kind of filtering out certain pathogens and things. But I think obviously we'll go through lymph node and everything like that. Tonsils are important. Galt, I mean, galt is important, but we'll go through it. Okay. Oh, wow, he just jumped right into immunity. Did I skip one on accident? Hmm. All right. All right, so starting with the creation of lymph, I think that's going to be an important thing to look at. So we look at, here's a lymphatic vessel. There's a blood capillary. So lymphatics and blood actually run pretty close together. If you look at like a simpler diagram, you can see the lymphatic capillaries and the blood capillaries almost interweave so they can actually share fluid. So kind of if blood is flowing this way, we're gonna have some pressures at work here. We're gonna have some hydrostatic pressure that actually pushes fluid into our extracellular space. And then this is where fluid can then also get into the lymph. So this is our hydrostatic pressure. This is coming from the left side of the heart. So when the left side of the heart contracts and pushes the blood systemically through the body, that hydrostatic pressure is pushing some fluid. So it could be ions, water, maybe some small proteins, maybe some pathogens, things like that into the extracellular space where it can then be picked up by the lymphatic system and then brought through the lymph nodes and some of the other lymph organs in order to kind of clean it out. But we always have to remember that if there's some pressure pushing stuff out, the blood is always going to pull some back. This is just like the kidneys. We're going to have some osmotic pressure. Now, osmotic pressure is due to big proteins in the blood. So you'll have things like albumin and other big proteins that will pull water back through. So even though the hydrostatic pressure is pushing some fluid out into the extracellular space, we'll still have some fluid being pulled back into the blood due to those big proteins pulling that blood or that water in. So this is how lymph is actually created. So when this gets into the lymph vessel, this will then run through, and if it reaches a lymph node, then we have a few structures I want you guys to know. So we'll have an afferent vessel. This means it's bringing it toward, so bringing fluid toward, and then we'll have an efferent vessel. This is pulling things away. So fluid will run in. Fluid runs out through the efferent. Now on the inside, we have some germinal regions. I'm just going to abbreviate them GR. And these germinal regions are where white blood cells chill and wait. So they could be markers for, they could have antigens or antibodies that are specific for certain pathogens that come through the body, or maybe they act as more of an innate response. Whatever it is, they're going to find pathogens and things that are flowing through the lymph and either phagocytose it to then present it to T or B cells, or like macrophages will eat it up, put it on their surface on an MHC1 or MHC2, and they'll work through it that way. So this is just kind of a waiting ground for white blood cells or immune cells, so we can filter that out. And that's how that kidneys work, or the spleen works too, right? The spleen has the white pulp. That's what germinal region. So when things pass through, it's going to be able to kind of catch things, catch pathogens and things like that as it's being filtered out through the blood. The other big one is the tonsils, which I know he spent a little bit more time on. So if we look at the tonsils, they have these things called tonsillar. I just think it's a funny word, but it's tonsillar crypts. The tonsillar crypts are where bacteria and stuff can be picked up and caught in, and then they can be transported across the cell or across the surface of the tonsil using an M cell. That M cell will pull those bacteria across, and then we actually have germinal areas that are hanging out by these crypts. So when we pull these in, we can then do what we need to do with that pathogen or bacteria. So usually the tonsils are the first stop for like food-borne things or things that you put in your mouth, since they're that first line of defense from eating. Any questions on lymph? This thymus really functions a lot when it comes to actually picking up T or picking out T's lymphocytes. So it actually does a selection process where it pulls T, and we'll go through this when we get to the immune system. So I guess we don't even need to comment on it, but they're going to be more for the T cells. React as filters, yes. White pulp, red pulp, all the things. Okay. Oh, the M cells, I wasn't very specific. So you have specific cells here called M cells. These are the ones that will transport I should just say foreign things across. So when I think foreign things, just think like bacteria, fragments of bacteria, other pathogens like that. So it will actually transport it across this barrier and then put it into the germinal region. So then those macrophages and things can look at it. It's a good question. I'm not sure if crypts are present in the intestine. You have your deep villi. But those gall regions are located more outside, like around like these areas. So I don't really know exactly how the crypts work there, or if there are crypts, but okay. Let's hit immune because I'm going to get stressed out about time. Do you guys have any questions on that? Okay, so let's do a quick flow chart because I love a good flow chart. So we're looking at innate versus adaptive. I'm going to add a few bullet points, then we'll put the cells. So the innate immune response is quick acting, while the adaptive is going to be slow. The innate is non-specific. So it cannot pick out a specific virus or bacteria and be like, I know what this is, I can fight it. It can destroy normal tissue as well. So inflammation and things like that, it can destroy very normal tissue that's not infected, which is why the adaptive is a little bit more specific in killing or getting rid of certain pathogens or bacteria. So the innate has two kind of things that we'll talk about. First one is going to be inflammation. Inflammation is due to bacteria. usually. And we have our a few cells at play. So our granulocytes. We have our neutrophils, our basophils, our eosinophils. Eosinophils deal with like nematodes, things like that. Basophils can do histamine release, which histamine is the one that plays that role in our cardinal signs of inflammation. And then neutrophils are all about free electrons. So they'll release free electrons that will kind of destroy everything around the bacterial infection or whatever it is. But that means those are also destroying normal tissue. So tissue we don't really want to actually kill off. The other cell type that I just like to add here is mast cells. These also do histamine. Cool. Now the other side of this will be interferon. And we'll talk about this when it comes to viruses. And really, interferon is a chemical messenger. From neighboring or self cells. So cells can do this themselves if they're infected. So if you have COVID and one of your lung epithelium get infected, then it can actually release interferon to tell the cells next to it that, hey, I'm infected with this virus. You should try to not get infected by the virus. So I want you to do all these things to not get infected, to try to have somewhat of a quick response to a virus. But these will be the two important ones when we talk about innate. And then adaptive is going to be all about our two branches as well. Sorry, my flow chart's getting wonky. So we'll have cellular and humoral. Humoral means kind of, it's back to that idea of when we talked about like hormones and things like that. Humoral will be things that can be actually secreted. So a lot of the big cell type here is going to be our B lymphocytes. B lymphocytes can become either two types of cells. They can become plasma. I should probably put naive. This is a naive B lymphocytes. These can either become plasma B cells, or they can just become memory B cells. Now our cellular branch is all of our T lymphocytes. And these will become either our T killer, or we could also call these our CD8 cells. And then we'll have our T helper, which would be our CD4 cells. So there's our two branches there. Questions on that? I do want to just add this because I like this little, I think knowing that MHC1 are the ones that will deal with T killer cells. MHC2 are the ones that will deal with T helpers. And I'll kind of talk about this when we draw it all out, but okay. Let's start with inflammation. I'm trying to decide if I want to draw a picture of this or just do it through steps. You guys want me to do picture or steps just for this one? It's kind of a lot. Okay, steps it is. So first step is damage to the dermis or skin. We always, every time every time I've learned this, we always talk about a splinter just because it is easy to picture. So a splinter, and I can kind of maybe just draw a quick picture on the side, but a splinter enters the dermis. So we'll have a splinter here. And this splinter has a bunch of bacteria on it. So first step, you have to have some kind of damage that happens, some kind of breakage to the skin. From there, bacteria will release cytokines. So some of the bacteria itself will actually release cytokines into that area. But the other big thing is that our fibrocytes, which if you guys remember back to unit 1, these are cells that live inside the dermis. These will also release cytokines. Cytokines are signalers, so they'll go off into the bloodstream and they're going to go signal things from the red bone marrow. So from there, maybe we'll add this as like a sub step. Cytokines go to red bone marrow and mobilize. What are the 2 cells that we're going to mobilize from the red blood or from the red bone marrow? Do you guys know? Yep, pattern associated and then damage associated. But what are like the 2 cell types that come in and start playing a role here? Yeah, the big one is gonna be monocytes. So we're gonna mobilize monocytes. We know monocytes become macrophages. So these are gonna come on over through the bloodstream and they'll start eating stuff up. But for now, we just want to think those cytokines are the ones that are immobilizing those monocytes, telling them, all right, we got to get over there and get working. The other one is our neutrophils. Remember, we're talking a lot about the neutrophils. Really, the neutrophils are going to be kind of your first line of action. Monocytes, because they become macrophages, they're going to come in and eat the bacteria, and then they can actually present a piece of that bacteria on its cell surface. And then you can actually have an adaptive immune response. But the neutrophils will mobilize as well. And then they're going to leave and enter that same area. So we can say 4 will be monocytes and neutrophils enter circulation. They'll make it to site splinter. And then what do the neutrophils do first? I think what's first important is that neutrophils use what's the name for the protein? Selectin, I think. Selectins. Yeah, use selectins to enter dermis. So neutrophils are too big to squeeze through the capillary, so they're going to utilize transmembrane proteins that are on the actual capillary or endothelial lining that they'll bind to, and then it's going to cause something called diapedesis, so that we can pull the cells through, and so they can enter the actual site itself. So now neutrophils release free electrons. These are going to start killing off everything around the area. So it will start to damage the bacteria, but in the same run, it's going to also start to damage the dermis itself, which is why this is not very specific and it can be harmful to the body a little bit. The seventh step, which I kind of skipped, it kind of happens early on too. This is all kind of happening at the same time, but our mast cells release histamine. This will activate the cardinal signs of inflammation. What are the 4 cardinal signs of inflammation?
Heat.
Perfect. Redness, perfect. Swelling, great. What's the last one? Pain. So pain is the only one that is not caused by histamine. This is going to be due to nociceptors or pain receptors, right? So we'll have some pain receptors on there, but not caused by histamine. All the other ones are caused by histamine. Because histamine will vasodilates blood vessel. Vasodilating mean we're opening up more of the blood vessel to get more blood to the area. That increases the heat. That makes it swell. That's going to also increase the redness there because we're putting more blood there. This also helps us get these cells that we need there, right? No, this all kind of happens at once. I know my numbers are kind of off, but cool. Last big one is we already kind of talked about, but the monocytes will come in. So monocytes will mature to macrophages and phagocytose stuff. I shouldn't say stuff. I'm getting lazy. Phagocytose bacteria. And then what's nice is macrophages are antigen presenting cells, so we can then present it on the cell surface, and then we can possibly have an adaptive immune response if we needed to. So can present on cell surface. Questions on that? The other thing that's released that he adds is heparins. Heparins help to also help with blood flow, thin the blood out. So heparin is a big like blood thinner. Doesn't allow it to clot as much, so you can get blood to the area. So I always leave out dendritic cells only because I think it is good to know. So I should say like know them, but they do the same thing that macrophages would do. So dendritic cells also will phagocytose things and put them on their cell surface. They'll help. Yeah, of course. Cool. Questions on inflammation? Okay, if not, let's do interferon. So interferon is neighboring cell to cell, and it deals with virus. So the example we always use is either the flu or COVID. I'll use the flu since that's one he talked about. But so right now we're in lung epithelium. So this is lung. Maybe it's actually pneumocytes in the alveoli. But think we're in the lung. We'll have an apical and a basilar side. So when a virus comes in, here's my virus. Let's say this is H1. Oops. H1N1. So this actually tells you exactly kind of what it is. So I hate this word because I can never say it. Hemagglutinin is going to be the required one for entry. So that's what that H stands for, is hemagglutinin. It's glutanin in whatever. So we'll have that receptor on our cell surface, that H1, and that virus will bind to it. Part of its proteins on that surface will bind. And then it's going to be phagocytosed. So it's going to be eaten up by the cell. Once in the cell, it can have some viruses work different ways depending on if it's an RNA versus a DNA, but it will essentially take over cell functions. The big functions are going to be transcription and translation. So we're going to take all those over so then this virus can duplicate itself. You can make a bunch of it. And if it can make a bunch of it, then it can actually exit the cell, right? It can be exocytosed. And it's going to need neuraminidase to do that, which is where we get the N1. So H1N1, this is our neuraminidase. Neuraminidase. And that's going to be required for exit. So now if this virus can exit, it can then go on over and infect another cell if it wanted to. However, this cell has a defense. So the defense is it's going to release what we call interferon. This would be IF1. So it's interferon type 1. I'm sorry, I like to use a lot of just abbreviations. So IF stands for interferon. I should really just write it out. Interferon. IF type 1. These will go to neighboring cells and find an interferon receptor, and it's going to tell this cell to do a few things, or these cells to do a few things. It's going to tell it to decrease transcription and translation, because we don't, if the virus were to get in, we don't want it to be able to take over the machinery. We want it to tell it to decrease all these so that if it does get in, we don't replicate it, it can't spread further. And then the other thing is we want to decrease endo and exocytosis. So we want to decrease how much we're putting out of the cell and bringing in. So if for some reason the virus were to bind to an H1 receptor on here, this cell would be like, I was told to decrease my endocytosis or my phagocytosis of eating this up, so I'm not going to do that. So then we can prevent infection. So that's kind of what the interferon's doing. It's working in an autocrine or a paracrine fashion. Where it's cells talking to neighboring cells, so we can prevent that virus from spreading. The other thing that this cell will do is the one that's infected can actually now place a piece of this, so it can take a piece of this virus. It can use the lysosome to kind of break it apart. It will go to the endosome in order to put it on an MHC protein. But we can also put it on the cell surface here, so then a T killer cell can come by and be like, hey, that looks like a virus that I should kill. So I'm going to kill the whole cell because the cell is infected. What type of MHC would I put on this lung epithelium? Is it MHC1 or MHC2? Perfect. 1. So MHC1 is always located on any nucleated cell. So it doesn't have to just be a antigen presenting cell like a macrophage. It could be any cell in the body could have an MHC1. MHC2 is specific for only antigen presenting cells, so B cells. B cells, macrophages, dendritic cells, cells that will present an antigen on it will have an MHC2, but they could also have an MHC1. Okay, that's my innate. Any questions on the innate?
What's the difference between a T helper cell and a B? Is it B plasma B cell, memory B cell?
Yeah, let's I'm actually gonna go through that. Let's do that next. So that kind of leads us on. So those are going to be our innates. Let's do our adaptive really quick. So adaptive immunity is going to be all based off of if we already had an infection and we were able to have a little bit more time in order to actually build a specific response to it. So starting with our humoral branch. These are going to be our B lymphocytes. So we have 2 types. Well, actually, we start as a naive B lymphocyte. And I'm going to draw this one because it's easier to draw. So we have a nucleus. Our naive B cell doesn't really know exactly what we want to become yet. So we need to be told what we want to become or what we need to become. Is kind of how this stuff is kind of how it's going to work. So antigens, so B cells are antigen presenting cells. Oops. APC, which means they can present an antigen or a piece of bacteria on an MHC class 2. They can also do a class 1 because they have a nucleus, but we're going to talk about it in terms of a class 2 since they can specifically do that since they're antigen presenting. So if this is my MHC1, or sorry, 2, MHC2, then we have an antigen on it. So it's found some sort of bacteria or virus that it was able to present an antigen on it. And now we can have a cell come in and help. And this is where a T helper cell comes in. So we can think of T helper cells as they're going to help B cells determine what they need to do, whether they become memory B cells or plasma B cells. So this will be my T helper. I can also call this a CD4 cell. And we call it a CD4 cell because it's going to have a little protein on it that's called CD4. This protein is going to help anchor to that naive B cell. So what's going to happen is this T cell is going to get closer and it's going to anchor. And once we anchor, we'll have another receptor here. This will be our T cell. Wait. Why am I losing my... Oh, no, I'm right. T cell receptor. The T cell receptor will, once that CD4 pulls that naive B cell closer, the T cell receptor will interact with that antigen that's presented on the MHC2. So you'll have some kind of interaction that happens here, and it's going to be like, all right, I recognize this as a foreign thing. We need to probably figure out what we want to do about it. There is another protein involved coming off of our CD or our T helper. We're going to have something called a CD40 ligand. And we'll have a CD40 receptor on here. Another way to kind of tell this cell, okay, this is what we need to do. At this point in time, the T helper cell releases interleukin. Let's do orange. So we lose interleukin 4. Interleukin 4 is a chemical messenger that is then going to come on over and it's going to talk to an interleukin receptor that's on the lymphocyte itself. So here's my IL receptor. When this binds, it's telling that B lymphocyte one of two things. It's either going to tell it to become a plasma B cell or become a memory B cell. Now that I look at this, let me just double check something really quick. When we have CD40 and CD40L, and we have interleukin 4, the cell type that we end up producing is going to be plasma B cells. Now, the difference between memory B cells and plasma B cells is plasma B cells will actually secrete their antibodies. Oops. So their antibodies will actually be secreted into the plasma and not held onto the cell surface. So we get our plasma B cells. Now, if I were to look at this, I'm just going to copy and paste this. We can do another one. And we just change the, essentially all we're changing is our, I hate that it does this. So if we change what this receptor is here, so instead of CD40, we're going to have a B7. I have to remember that. And then we'll have a CD28 on the B cell. So then these two receptors will interact. And then this is going to cause, instead of the release of interleukin 4, we'll have the release of interleukin 2. Interleukin 2 will come over. It will bind to an interleukin receptor. What is happening? Sorry, I'm checking something just to make sure I have this in the right track. Because you can do either T or B. Okay. So in this case, the interleukin 2 works specifically on the T cell itself. And then we end up with clonal expansion, which stands for essentially just differentiating to get more T helper cells. So then we can end up getting more. So we activate B cells, but then we can also activate T cells. So we'll activate B cells and T cells. So we'll end up with getting more T cells. And then we'll end up getting more activated B cells. So if we're looking at this, the other option we can actually get is what we call our memory B cells, which will be cells that have antibodies on the surface. Do you guys have any questions? I feel like I just got myself confused. Or correct me if I'm like missing something that he might have said. So memory B cells hold antibodies on their surface. So that would be these guys. So instead of putting them out into the plasma, they'll put them on their surface. So that's kind of the role of the B cells or the T helper cells with B cells. Now the other role is going to be the whole idea of T killer cells, which T killer cells are going to work on MHC1s, which means we can work on basically any cell that we can. So if we have, let's just say we actually come back to our example here. So we had our H1N1 attack this epithelial cell. So what we can do is a T killer cell can come in and it will have very similar proteins. So you'll have a T cell receptor. This will interact with our MHC1. We will have a CD8 protein that helps us with our anchoring. And then this is going to cause, so once this actually interacts, we will have the release of, so what will happen is the T killer will realize, okay, this cell's infected. We need to kill it right away before it kills off any other cells or that infection can spread. I meant to label this T killer. So we're going to relieve what we call peripherin. Peripherin is going to be a molecule that will just directly either proliferate through the cell membrane, and when it proliferates the cell membrane, it's going to allow calcium to enter the cell. Because the calcium gradient is higher outside the cell than inside, calcium is going to want to enter the cell anyway. So if we blow a bunch of holes into the membrane, calcium can then come in, and then that's going to cause cell death. So the increase in calcium entrance causes cell death. And that's how that T killer cell is working. Does have any questions on any of that? Cool. All right. Let me see if there's any other quick questions on here. Activate the lymphatic system. The T, I'm just going to very quickly say this, but T cells are all due to the thymus. The thymus does two things. It looks at T cells, and if the T cell recognizes proteins, then it's good to go. It can move on and do its job. If the T cell does not recognize proteins, then the thymus will tell it to go and die. So basically it's going to be like, you're not functional enough to actually go work. Sometimes some of these T cells, if they can't recognize foreign proteins, but they can recognize self proteins, then they can end up causing autoimmune disorders where they start breaking down our own proteins in our own cells, things like that. So sometimes the system doesn't work perfectly, but it usually uses that T cell, or the thymus is going to look at those T cells in order to send it on to the next step. Okay, that's a lot. Any questions on that before I move on to digestion? I'm definitely not going to get to all these key questions, so I'm just going to do digestion pretty quick. Actually, let me ask your opinion since we only have like 40 minutes. I am going to either upload a video of me going through one of these, like without you guys having to sit here through it. Do you prefer me do digestion right now for the last 40 minutes or do urinary? Urinary. Okay. We can do urinary. Okay. Awesome. So urinary, pretty straightforward actually. So if we think about just an anatomically thinking about the urinary system, let's look at, we'll just draw one side. So kidney, obviously important. Coming off, let's just do all of the anatomy really fast because that will help us for later on. So the kidney has a renal artery, has a renal vein. When we look at the kidney itself, we're going to have a cortex as kind of the outside surface or the outside area. And then we're going to have what we call the medulla. So we'll say this is the renal cortex. And then we'll have some, as we get deeper, will be the renal medulla. So I want you to think that I like cut this kidney. So we're looking at it like the other way. Inside here, we'll have pyramids. Call them. And these dip down into that medulla. These are my renal pyramids. Coming off each pyramid, we'll have a renal calyx. And then this all empties into a big thing called the renal pelvis. And from there, these empty out into our ureter. So when we're making urine, essentially it's going to start to build up in the pelvis, or from the calyx to the pelvis into the ureter, and then that's going to enter down into our bladder. Bladder. And then coming off the bladder is we have our urethra. And this is really where it changes depending on male or female. So female, or if you have the uterus, then your urethra is a lot smaller, and it's essentially just going to enter the outside world pretty quickly. A male, they'll have two sections of their urethra. So their first urethra is around something called the prostate. So this is our you can call it the prostate. It's not prosthetic. Prostatic urethra. So where it goes through the actual prostate itself. and then we'll have the rest of the urethra that runs down through the penis. And then obviously the external part of it is on the end, or the glans. Penis is the end of the penis. That's where you're going to have the end of the urethra. So we have a lot longer urethra, or males, if you have a penis, you have a lot longer urethra than females. They're going to have a lot shorter one. So that's general anatomy. Just kind of important to know. So now if I zoom back in and look at our kidney, what's important to note is that when the artery comes in, it's going to start to branch into smaller and smaller little arteries. And there's a few that I want you to know when we get to a smaller diagram, but we are going to talk about an afferent and an efferent arteriole. Those are really where we get down to that glomerulus, which is a specialized capillary. So sitting around the cortex, we have our nephrons. So we'll have a Bowman's capsule, we'll have a descending limb, an ascending limb, and then we'll enter a collecting duct. I'm going to draw two types of nephrons here. This nephron is what we call a juxtamedullary nephron. It's going to dive all the way into the medulla. So it's going from the cortex to the medulla. You'll have different length nephrons. So you could also have a cortical nephron where it stays up here and it doesn't go all the way down into the medulla, but it just stays a little bit higher. So you have a few different types of nephrons. I think the most important one that you guys really need to know is the juxtamedullary nephron. This is the one that really dips all the way down into the medulla, and where we actually have our corticomedullary gradient and all of that fun jazz. Questions on that? Okay. If not, let's jump right in. Let me just look at the difference epithelial found in the histological. I'm not going to have time to go over this, so make sure you go over two. Bladder is transitional epithelium, or transitional squamous. I don't remember. Transitional so that it can actually inflate and then bounce back. Look at where sections in the urethra, which ones are stratified squamous versus simple squamous. All that stuff. Again, don't have time. I'd rather go through the other stuff. Okay. Let's look at the nephron, since that's the functional unit of the kidney. So that's really where we're actually filtering our blood, filtering things out. Make sure you go, I'm looking at some of the slides, just make sure you go over some of the like UTI stuff like that. Okay, so let's start with just a nice diagram. So we have our afferent arteriole. So this is bringing blood towards the nephron. From here, we have a specialized capillary. This is called a glomerulus. This is where the blood is actually being filtered out. So the glomerulus will actually have some bigger gaps between the cells, which we call fenstration or fenestrated. Fenestrated meaning window. So it has a little bit more of a gap so that fluid can actually be pushed from the blood into the nephron. And we're not trying to squeeze through a tight gap. And then we have our efferent arteriole, which is where the blood has now been filtered. Oops. So now we're going to have filtered blood. Okay. And surrounding the glomerulus is our Bowman's capsule. We have specialized cells here called podocytes. They're also slightly fenestrated with a bigger gap, so we can let more fluid into the actual Bowman's capsule itself. So this actual structure is called Bowman's capsule. All together, so with the glomerulus and the Bowman's capsule, this is our renal corpuscle. From here, we enter a region called our paroxysmal convoluted tubule, or PCT. Then we'll have a descending limb. This down here is our hairpin turn, or we could also call this the loop of Henle. We'll have two parts of the ascending limb. So I have a thick and a thin. So here's my thick segment, and here's my thin segment. And then our last section is our distal convoluted tubule. And then the collecting duct is the last part, but the collecting duct is not actually considered part of the nephron. So it's like its own little thing here. This is our collecting duct. Oh, okay. So kind of starting at the top here, let's go through some of the pressures that we have going on. So like we talked about with lymphatics, the pressure that comes from the left side of the heart is going to cause something that pushes fluid from the blood into Bowman's capsule. Oh, shoot. I don't know what I did. Slid it. So the orange arrow is going to dictate our glomerular hydrostatic pressure. And this is usually pretty high. It's around like 55, I think, millimeters of mercury. So that's the pressure coming from the left side of the heart. So when the left side pumps, pushes that blood systemically, it's going to push blood from the glomerular, or not blood, filtrate, or like, ions, glucose, amino acids, proteins, things like that into the Bowman's capsule or into the nephron at that pressure. But we will also have two pressures that kind of push back on this. So we'll have one that pulls water back. And just like we said in lymph, this is going to be called our blood colloid osmotic pressure. This is just a really fancy way of saying the pressure that's caused by blood proteins. So proteins that are in blood, like albumin and other big proteins, pull some water back, just like in lymph. So we'll have some water being pulled back. 30. And then our last one is we'll have a little bit of hydrostatic pressure also coming back into the blood. So this would be tubular hydrostatic pressure. And this is just pressure due to how fast the fluid is entering the nephron, it bouncing off the tubule wall. So like it bouncing off here will then push some of the fluid back into the blood. And that's pretty low. It's not high. So we'll say it's like 10 millimeters. So we can calculate this if we were to take 55 as like the total pressure coming in, but then we would want to subtract it by the two pressures that are pulling stuff back into blood. So we would have 40. So I'm not good at math. 15. So that's 15, which I think his example was 10. Again, don't stress too much about values, but let's say we just have 15 net pressure moving that way. In your glomerular filtration rate, which is what this is like the pressure that fluid is running through that area or that nephron, can change depending on your blood pressure and things like that. So if things are moving really fast through the kidneys because you have high blood pressure, then you don't have a lot of time to reabsorb stuff. But now I like to break this down into what's being absorbed and secreted in each section. So, and you know what? To save time, I'm just going to jump over to a picture that I already had drawn of this, just so we can save a little time. Okay. So talking about the PCT. So the proximal convoluted tubule. We have a few things that are going on here. First big one is we need to absorb a few really important things. The first big one is we need to absorb our glucose. And we need to absorb our amino acids. We don't want to be urinating or getting rid of glucose or our proteins. So these are two big things we want to be able to take from that was put into the nephron, take it out of the nephron and put it back into our blood. Other things will be ions. So we'll have some sodium, some chloride. Water will follow it because water always follows salt. So we'll have some water pulled back in. And then we'll have some other things like urea and things like that. So if we look at kind of the overall picture here, we'll have some other small proteins, sodium, potassium, chloride, calcium. Amino acids and glucose are the big ones. You usually will have 100% of these things reabsorbed back into blood from here. Things that are put into the nephron from here can be drugs. So some of the drugs that we take are excreted through the kidneys, so we can have some of it secreted back into the nephron. Urea, uric acid, some acid, creatine, which is something that comes from the muscle. So all of this can be put into the nephron and filtered through. But again, we really want to save the amino acids and the glucose, which is why this drawing really just dictates those two things. So with amino acids and sodium, I want to dictate this is inside the nephron. So we're inside the lumen of the nephron. This will be outside. And then we're going to end up having a blood vessel right here called the peritubular capillary. This sits right next to the PCT. So it's really close in proximity. So we can really take whatever that glucose is or that amino acid and pull it into the blood directly from the nephron. So if we're looking at this, we need a glucose sodium co-transporter. So know what a co-transporter is versus an active transporter versus a one that pushes things opposite. So co-transporter means we're going to need sodium and glucose in order to open this transporter. Or glucose is going to, in another way of saying it, glucose will need sodium to let glucose into the cell. So these are going to be located on the inside of the nephron. When it senses that glucose, a sodium and a glucose will bind to this channel and you'll transport both into the cell. So that's what this red guy is here. We've transported the glucose into the cell. And from there, it can actually leave the cell off the basilar side through what we call a GLUT channel. It's a facilitated glucose transporter. So it's only transporting glucose. We don't need sodium anymore. We don't need any of that. We can just pull the glucose from the cell and put it right back into the blood. Now for the sodium, to get that out, we're going to use, and it kind of when in doubt in the kidney, we're always going to see a sodium potassium ATPase. ATPase tells you we use energy or use ATP. So it's going to be an active transporter. And that's going to help because we're pushing sodium against its gradient. Sodium's higher on the outside of the cell. If we're trying to get sodium outside of the cell, then it's going to be harder to push it that way because it doesn't want to go. So this is going to push once, I think it's 2 sodiums and then 3. You guys can correct, you can look up the little details. I think it's 2 and 3. 2 sodiums out, 3 potassiums in, or it's the opposite, whatever. But that's how we're counter transporting. So we're pulling 2 things against their gradient. So we need that energy to do so. And that's what gets that sodium here outside of the cell. So then it can possibly enter the blood or it can just stay in the extracellular space. Now, amino acids are going to use the same thing. So if we had an amino acid here, they need some sort of facilitator too. So we're going to have a channel specifically an amino acid sodium cotransporter. So if an amino acid needs to be let in, we'll also bring in a sodium. That sodium will then go and use an ATPase to get out. And then now we'll actually have a specific channel on the basilar side that is an amino acid facilitated transporter. So now we can take that amino acid and put it into the blood. Now, with all this movement, water is also going to want to move, and water actually doesn't move through channels. It's going to move between the cells. And we call this paracellular transport. It's just a fancy way for saying moving between the cells. So instead of through the cell, it's going to go between them. So if you remember from unit 1, we have tight junctions that hold the cells pretty tight together, but water can kind of slip through there. And if we have sodium and other ions moving that are positively charged, it's going to pull some of that water over as well. So we're going to have water moving kind of the whole time. A term that we use for like the glucose to come through, because it's moving through the cell, we call this transcellular. So anything that moves through the cell is transcellular. Anything that moves between is paracellular. And those are kind of the big things when it comes to PCT. So again, we want to really focus on pulling in all of that glucose and amino acids so we're not urinating that out. Usually if you see this kind of stuff in your urine, then that means you have some sort of dysfunction going on, whether it's in the kidneys or somewhere else. So they can test for these things in the urine. They can look at it. Questions on PCT? Awesome. Descending limb, super easy peasy. Descending limb is all about water. So water is actually going to move through channels this time though, so not through. And they move specifically through what we call an aquaporin. Aquaporin is just a specific channel for water. Aqua, you can kind of just think of it in the name. Aquaporin, it's a pore for water. And it's going to put water directly back into the vasa recta, which is the blood vessel that surrounds the descending and ascending limbs. And when we get to this idea of the corticomedullary gradient, where things get saltier as we get closer to the medulla, this water idea will make more sense. But I think just at least looking at this as the only thing that moves is important. So water is coming from the inside and being put back into the blood. Ascending limb, also fairly straightforward. We're going to have passive ion channels. So ions like potassium, chloride, sodium will passively diffuse. So they'll have specific channels that are for that ion itself, but they'll passively diffuse because they're going to be moving with the gradient. Since we get less salty as we come up, we're going to have ions wanting to passively move across the cell surface. So these are moving transcellularly, so through the cell. But ascending, or the thin segment specifically, the thin is passive. Now the thick segment is a little bit different. We do need a cotransporter. So we're going to have a sodium potassium chloride cotransporter. So it's taking a sodium, a potassium, and 2 chlorides at the same time and pulling them into the cell. And then we'll have, again, that lovely ATPase comes back, and it's going to be used to then move these around. So we pull the sodium out, pull some potassium in, and then you'll probably have some movement of the other ions as well. But the thick segment is a little bit more regulated because we've gotten less concentrated. As we move up into the cortex, we have less salt on the outside. So now we don't need to let go of more ions, and we have to kind of facilitate how we're moving them more. And then the last one is the DCT. This is like my big saying with the DCT, is its last option for sodium. I can't spell. Sodium reabsorption. So it's the last place that we can really pull in sodium if we need it. So we have specific cells called macula densa cells. These macula densa cells actually have a sensor on them that it senses sodium chloride concentrations. So if concentrations are high, so high sodium, high chloride concentrations, that means we're moving too quickly through the nephron. We have the filtrate that came from the blood is moving way too fast. We don't have time to pull sodium out. So at the end of the nephron, we're like, oh shit, we have a lot left. So what we want to do is pull more back into the blood. And what we're going to use is something called adenosine. Adenosine is going to go act on smooth muscle that's around our vasculature. So we have smooth muscle that's around the afferent arteriole, not the efferent. We want to constrict the afferent so that we can slow the blood flow down a little bit. So we'll have vasoconstriction, which will decrease the GFR. That should say GFR, glomerular filtration rate. If we're slowing down the filtrate rate through the kidney, then we can actually, or through the nephron, then we can really decrease, or we can increase the amount of sodium that we're pulling in. So by the time we get to the DCT, we can be like, all right, we've pulled enough sodium and we can let some of it go. Our body doesn't need any more, but we have a good GFR rate. So that's one way the DCT really works to like help regulate certain things. Another thing is it's going to also work with aldosterone and other hormones like that, which we will get to here in a second. But they're going to be the last ditch effort for sodium. So the big one that I drew is just sodium. We have very specific channels called ENaCs. I don't know if he brought these up. I love the name because it's just epithelial sodium channels. Literally is just like a fun way of saying epithelial sodium channels, ENaCs. But pretty straightforward there. Co-transported, Jess, sorry, missed your question. It's co-transported with potassium and sodium. Okay, so that's your DCT. Obviously, the last one is our collecting duct, which I have up here. Collecting duct is also through aquaporins. It's the last place that will pull in water, and it's regulated by ADH or vasopressin, which comes from our, where does the ADH come from? I spelled vasopressin so wrong.
Posterior pituitary.
Beautiful, yes. It's one of the only ones. What's the other one that comes from the posterior? That's a great final question.
Oxytocin.
Awesome, great. Okay. So these are controlled by ADH. ADH will tell us whether we need to pull in more water or not. So a lot of the time, this is the one I think it's a great example. If you drink too much one night, alcohol suppresses ADH. So instead of pulling water into the body, we're letting water go through our urine. So you're peeing more frequently, but then you're also dehydrating your body because you're not pulling anymore since we've inhibited that. I think it's just a great example to think about when you're like thinking about this specific hormone. Okay. Okay, so let's talk about the medullary gradient because that's important. And you know what? I'm going to go to a specific slide because I actually love the picture of it. And to me, it makes so much more sense. This slide. Okay, so if we're looking at just everything on here, as we get deeper, so this is our renal cortex. So when we looked at the kidney, here's our cortex, right? Is the outer side or the outer edges. The medulla dips low and dips into the deeper part of the kidney. And this image is good too because it kind of shows in retrospect these values. So at the cortex, we're at about 300 millimolars of salt concentration. Not very much, it's low. As we dip deeper into the kidney, we get all the way up to 1200 millimolars of concentration. And when I say concentration, I like to just think about saltiness. So think that as you get deeper, it gets more salty. When you're up here, it's a little less salty. So now if we're looking at this image, we're starting in the cortex. So the glomerulus and the renal corpuscle, oh, it says it right here. The renal corpuscle is all up in the cortex area. So we don't have a lot of salt out here. It's not very salty in the outside environment. So we don't have a ton of movement of sodium. We don't need a ton of movement of water either. Water will move paracellularly because some of the other ions and the nutrients, but this is more about the glucose and amino acids that are moving. So that stuff's leaving and entering the blood here, glucose, amino acids. Now, as we start dipping, we increase the osmolarity, and we're increasing that because we're increasing salt concentration out here. So out here, we get more salty. And if we get more salty out here, the way to fix that is we pull water out. So in the descending limb, we have aquaporins that are pulling water out of the nephron into that salty environment because we want to try to equilibrate the inside of the nephron in the outside environment. So as you can see in the nephron, we also got really salty. You can see it with all the little black dots. Just think of those as like ion concentration. So we get more concentrated. So when we look at the hairpin turn, this is going to be the highest concentration of solute. Oops, it's not really working. Highest concentration is down there. So it's pretty salty at the bottom there of the hairpin turn and on the outside, and we want those two environments to match. But now as we start to ascend, we're now losing saltiness, right? We're getting less osmolarity. So now we want to pull ions out. Now we're like, wait, we were so concentrated down here in the nephron that now we need to pull some ions out of there and put it in the extracellular space. And then we can become less concentrated up here once we get to the DCT, which the DCT is that last place we can do sodium. But at this point, if we feel pretty confident and we're nice and low in concentration, then we can just let this go if we wanted to. And now at the end here, we'll concentrate the urine one more time because we're moving back down, right? So as we're moving back down this gradient, we're pulling water out because we want the inside of here to equal the outside of here. And if out here is really salty, then we're going to concentrate the urine again and then get rid of those ions on the outside. So that's how that medullary gradient works, is as we're going down, we're pulling water out because we're getting saltier and we want both the inside of the nephron and the outside to be pretty close to equal. And then as we're coming up, we're getting less salty. So now we want to pull ions out into the extracellular space so then the inside and the outside can be equal. Same with the collecting duct. We drop back down, so we pull water out. Now, the other factor to this is that we also add in our vasa recta. The vasa recta is the blood vessel that runs counter current, so it's going to run the opposite direction. So filtrate is running this way. The blood's running the opposite way. So it's going to kind of do the opposite. On this side over here, it's going to pull in that sodium that we pulled out of the nephron. That sodium chloride and those ions, because it also wants to be in equilibrium down here too. So it's also going to get really concentrated and salty at the bottom of the hairpin turn. We pulled in all of that sodium and all those ions that diffused passively here. But now as we start to ascend the vasa recta, we pull in the water that we just had because now we need to offset how many ions we just pulled into the blood. And we're getting less concentrated as we come back up. So now we're going to pull the water in that we pulled out of the nephron into the blood. And now we've gotten back to a lesser concentration. It's a lot of words. Do you guys have questions on any of that? Takes a second to fully understand, but I think if you can really look at just how it, and it's just the black dots to me make so much sense. As you get lower, we get a lot more concentrated, a lot more dots. And as we come up, we get less because we pulled some out, but we also are less concentrated at the top. So we don't want the nephron to also be super concentrated. Now, last thing I want to show you really quickly, and then we'll just walk through it and you can take a picture of this if you want, is this Renin angiotensin system. So we kind of talked about this when it came to the hormones, but this is really important when it comes to the kidneys because it directly affects the kidneys. Renin is actually released by juxtamedullary cells that are in the afferent arteriole. So if the afferent arteriole, which is where the blood is entering into the glomerulus, if it feels like it has mechanoreceptors. If those receptors feel less stretch on the artery, then it's going to be like, oh shoot, our blood pressure is dropping. We need to increase our blood pressure. So renin is released from those cells. From there, renin is going to act in the liver. It's going to be converted into angiotensin 1 in the liver. In the lungs, it becomes angiotensin 2 using an enzyme called ACE, which is an angiotensin converting enzyme. So we convert it from 1 to 2. And then that last step is it's going to cause vasoconstriction. So angiotensin 2 specifically causes vasoconstriction. So we're going to constrict our blood vessels to increase blood pressure. And then the other thing it's going to do is it's going to cause aldosterone release from the adrenal cortex. If we remember, the adrenal cortex sits on top of the kidneys. It's a steroid hormone, which means it's going to enter the cell directly. It doesn't need a GPCR. It's going to go right through the cell membrane. And it acts as a transcription factor, and it's going to increase ENaX. So it increases the amount of epithelial sodium channels that we put on the cell surface. If we increase the amount of ENaX, then we can pull more sodium into the blood. And if we pull more sodium into the blood, it's going to increase blood volume. And if we increase the amount of volume in the blood, then we can increase blood pressure. So kind of the end all be all is that we're increasing blood volume to increase blood pressure. So in an emergency where we're bleeding out or something, this can work to pull in some sodium, which if we pull in sodium, water will follow. So that will also increase blood volume and so on. But that's one of the mechanisms. The other mechanism is obviously ADH. If we are bleeding out for some reason, or we need to concentrate our urine, whatever it is, or dilute our urine, so make it more watery. Concentrate our urine. I said it opposite. If we need to concentrate it, so pull out more water, or we're dehydrated, then our posterior pituitary will release vasopressin that will run through the blood. It will bind to a GPCR. Vasopressin is a peptide hormone, so it can't go through the cell. So it will bind to a GPCR, and it's going to also upregulate aquaporin activity. So then we increase the amount of aquaporins that are exocytosed, increasing the channel activity, pulling in more water, which could also increase blood pressure if needed. Questions? I ran right to the end. Sorry I didn't hit digestion. This is like so much information, so hopefully that helped. I'm going to end the recording. Do you guys have